Translation process. The first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA, a process commonly called as charging of tRNA or amino acylation of tRNA. See in the tRNA diagram at the 3' end, there is amino acid. Very important. The cellular factory responsible for synthesizing proteins is the ribosome. These are non-membrane bound organelles found both in prokaryotes and eukaryotes. Now, the ribosome consists of structural RNAs and about 80 different proteins. So ribosome is made up of structural RNAs and 80 different proteins. Now, in its inactive state, it exists as two subunits, a large subunit and small subunit, that is 70S and 80S. We know in respectively prokaryotes and eukaryotes. Now, when the small subunit of this ribosome encounters an mRNA, this small subunit, the process of translation of the mRNA to protein begins. Okay. And there are two sites in the large subunit so that the subsequent amino acids bind and they make the formation of peptide bond between them. Now another thing about ribosomes, they also acts as a catalyst, that is 23S, a part of RNA which ribosome is made up of. The 23S RNA in a bacteria is the enzyme, that is ribozyme, for the formation of peptide bond. So this was it. Another thing in the section of methodologies in the human genome project, the methods involved two major approaches. One approach focused on identifying all the genes that are expressed as RNA, referred to as express sequence tags, ESTs, and another one is the sequence annotation. In that, it's a blind approach of simply sequencing the whole set of genome that contain all the coding and non-coding sequence and later assigning different regions in the sequence with their functions. So this is sequence annotation. The sequence of chromosome 1 was completed only in May 2006. This was the last of the 24 human chromosomes, 22 to be autosomes and XY to be sequenced.